Hi guys, Dr. B here. So this is chapter 9. Um, it's a lot in this chapter, but there's a lot of things in this. However, I'm going to focus on certain ones, certain items from here, and those are the ones I really want you to pay attention to. That's a really good hint for y'all, okay? I will do my best to point them out. Please pay attention to it. All right, so what do you think were the leading causes of death in the 1920s? So think about this is before like all the medical advancements we've had. So the leading cause of death, before vaccines and everything was actually infectious diseases. So infection. However, now, it would actually be cancer and heart disease. These are the ones that are more common or the leading causes of death. And why the change? Because this we learn more about how to treat them, and one of the ways we do that is through antibiotics. So this is just kind of a thing to kind of introduce this chapter in terms of DNA and all that. But this is just information that you should be aware of as well. So we're going to talk about transformation. And if you know me, you know what's going to come up in 2 seconds. And no, it's not this kind. One of my favorite scenes, by the way, from the Bumblebee movie. Sorry, that's the sound that they make when they transform. Anyway, moving on. So, Frederick Griffith, okay, 1928. This guy, he was trying to prepare a vaccine against pneumonia, a type of pneumonia caused by Streptococcus pneumoniae. That's the name of the bacterium that can cause this type of pneumonia. He used two different strains. One was rough and smooth. So the smooth strain has this capsule on it, and it's protected from the host. So whatever they're trying to infect, they're protected because they have this capsule on it. That means the host will die. The rough strain does not have it and is therefore killed by the immune system of the host, whoever they're trying to infect. So in his experiment, bacteria from the smooth strain were killed by heat, and the remains were added to the rough strain bacteria. And with this here, what happens, he is able to kill the mice because he did something within the smooth bacterium. He was also able to get some type of rough and smooth strain from the dead mice, and he concluded that there's something that had transformed the rough strain into the smooth strain. There's some kind of transforming principle, hence the meme earlier, right? Or the GIF, I'm sorry. So what we know now is actually the transforming principle was the DNA. While the bacteria had been killed, the DNA survived the heating process and was taken up by the rough strain bacteria. The smooth strain bacteria contains the polysaccharide that protects the bacterium from being affected by whatever it's trying to infect. So just looking at this, best way to describe it. So the rough strain injected into the mouse, and the mouse lives. You have the smooth strain, and then that mouse dies. So if you have the smooth strain, you apply heat to that, you actually will affect something. So you're affecting the DNA, and all of a sudden the mouse lives. Checks. A mixture of both rough and heat killed smooth. So the rough strain and the smooth strain that was affected by heat, the mouse here dies. Oswald Avery, in 1944, Avery demonstrated that DNA is responsible for transformation. Hershey-Chase experiment, 1952. Sounds like a chocolate. Anyway, so they used a bacteriophage. A bacteriophage is actually a virus. A bacteriophage actually will infect a bacterium by injecting its genetic material into it, which you can kind of see in these images. So black and white image, really, really small. This is a really small image. It's really magnified, I should say, actually. So this is what it looks like under an electron microscope. You have the head, core, sheath, and then you have tail fibers. So what they did was they labeled the bacteriophage DNA with phosphorus 32. So this form of phosphorus is actually radioactive, so they can detect it. They then followed the phages while they infected E. coli. They found that the radioactive element was only in the bacteria and not in the phage. In the second experiment, they put labels on phage protein with sulfur. After the phage was attached to the bacterium, the radioactive element was found in the phage but not in the bacterium. This showed that the genetic material which infects the bacteria is DNA. So what this means is that when they did the experiment, they could not find whatever they labeled radioactively. In the second experiment, it was still found within the bacteriophage, within the virus, but not in the bacteria. However, the radioactive phosphorus in the previous experiment, when they attached it, to the DNA of the bacteriophage, they found that actually it was in the bacteria. So that means that whatever is injected that infects the bacteria, that's the one that's what the DNA was. So this is again showing. So this side right here, they labeled the protein capsule with sulfur, and they can see that it's still not part of the E. coli. They labeled the DNA core with phosphorus. Whatever they inject in here was actually found in the bacterium and not in the bacteriophage after the fact. So the structure of DNA, a double helix and a set of nucleotides, which we'll get in more detail about. So what is a double helix? So a double helix refers to two strands twisted around each other, kind of like a winding staircase. You'll notice that it contains a backbone and all these little things right here, all these little base pairs in there, if you will. So we talked about the double helix. Now we're going to talk about nucleotides. A nucleotide is a subunit that makes up DNA. It has three different parts. It has a phosphate group. It has some type of sugar right here, and also a nitrogen-containing base. So in DNA, this sugar molecule is called deoxyribose. And actually that's where the name DNA comes from. Deoxyribose nucleic acid. So while the sugar and the sugar molecule and the phosphate group are all the same for each nucleotide, the nitrogen bases can be one of a few kinds. Star. Purines are adenine and guanine. Pure as gold. Please just keep in mind that I'm doing this for you guys, just please. Next one is the pyrimidines. So the uracil right here, this is actually related to RNA. So pyrimidines are cytosine, uracil for an RNA, and thymine. So pyrimidines, right here you see pi, sort of. So how do you get a piece of pie? You have to cut the pie. So this is just something that it helps me remember it, and so cut a pie and pure as gold. So here is just a quick little, diagram of DNA. You can see, what do you notice here? That you have hydrogen bonds in between each of these nitrogenous bases. So you have 3 hydrogen bonds between G and C, and then 2 hydrogen bonds between T and A. And I will discuss a way to help remember this, but I won't do it for this video.